Good morning, everyone. People are coming in. Hello.
Good morning.
Hello.
Good morning.
Good morning.
Good morning.
That's a slow start today.
Yeah.
It should be forbidden to come to work with this weather conditions.
Yeah, that's what I'm doing.
Yeah, I haven't done that. I regret it.
Did you get soaked?
Completely, like entirely soaked. I can when I squeeze my pants, water comes out of my pants. I'm not joking.
Surely someone told you that winters in Bordeaux are very wet and you should get raincoats.
Nobody told me. I thought that's why I don't have even a winter jacket, because I thought it's going to be 30 degrees throughout the year.
No, don't listen to them. Every year they say, oh, this is an exception. But it rains every year in the winter, non-stop.
Yeah, between October and December, it's terrible in Bordeaux.
Yeah, I see. I'm escaping for December, like throughout December, January.
Try London, guys.
Is it raining for you too, Chris?
I mean, it never stops.
But you're quite negative about the weather in London. It's not actually that bad.
I mean, you would see the mud everywhere around our flat. You wouldn't say that. I have to walk through the mud to go to work every day.
I see. Well, who's presenting today?
I am presenting.
Great. Good to hear. In that case, it's 9:32 now. So welcome everyone to Neuroccino, our weekly online journal club. Everyone is welcome to join us, discuss a paper, a book chapter, or whatever piece of science they find interesting. And the way it works is we just come online, discuss a paper, and to the best of our knowledge, and that's pretty much it. Just not your own work. That's the only rule. Anna, if you want to share the thank you in the chat. And I'm going to put that over on YouTube as well, where people can also ask their questions. And then just share your paper in full screen, please.
I am sharing. Can you see it in full screen?
Perfectly, yes.
Awesome. Cool. So welcome back to Neuroccino. Today it's going to be a review I'm presenting. So we're going to talk a little bit about evolution and the evolution of central neural circuits, the state of the art and the perspectives. So it's not going to be something that we can discuss, where we can discuss the methods in the end, but maybe some insights from evolution. So the paper was published about 3 weeks ago, and it's a super good read. I won't be able to go through the entire paper because it's just beyond the scope and the time limits of Neuroccino, but I recommend it very, very much to go through it yourself at the end. So it's by Roberts, Pop, and Prieto Godino. I hope I'm pronouncing it correctly. And they talk about principles of the neural circuit evolution, since the majority of the behaviors that animals, and us included, show arose through the evolution of neural circuits. And because of the technology that we nowadays can access, the neural circuits have been made more accessible, and breakthroughs have been made possible through through cross-species genetic modifications and through connectomics that we also are a little bit involved in, at least, and transcriptomics. So that these all things have been made possible to study different animals, different organisms on a comparative level. And the use of these different diverse organisms has also broadened already our perspective in neuroscience, and also in the just general understanding of how brains develop and function. So this paper basically merges the evolutionary biology and neuroscience, and they dive into kind of creating links between neural behavior changes during the evolution. what they do here is that they review examples of studies of species that have just given important insights into the mechanistic basis of the central neural circuit evolution. And they do focus on three main aspects. So it's, you can't see it in completely full screen, the entire picture, right? Oh, you can. Okay.
Oh, we can.
I will go through the diagrams in a second, but what they actually focus on are three aspects. So it's the synaptic connectivity. That's what you can see here. So they dive into the changes in neural circuit elements and focusing on the connectivity, on neuromodulation, and on neurons. So what I'm going to focus on today is the connectivity, since it's the most, the closest thing to our group, and maybe I am hoping to have interesting discussions, at least a little interesting discussion about it afterwards. The neuromodulation part and the neurons part is also super interesting, but as I said earlier, it's a little bit beyond the time limit. So what the authors do in the beginning is that they address a couple of questions that kind of guide them through the entire review. The questions are, let me find them so you can, it's so big here on my screen. I'm sorry for scrolling here through it. Oh yeah, here we go. So they address some open questions in the circuit evolution and let them just guide, use the questions to guide them through the entire review. So the first question that they address is whether the sensory periphery is more evolvable than central circuits. And they say that there have been a bias eventually, but still it's like not really clear whether the sensory periphery is more evolvable. So they're gonna address that in every aspect they dive into. So synaptic connectivity, neuromodulations, and neurons. Then the second question they try to answer in the review is whether some evolutionary change is more frequent than other evolutionary change. Also, they look into the hotspots of neural circuits. So they look at whether there are kind of central spots that are targeted by the evolution. And lastly, the question 4 they're guided by is whether all central circuits evolve according to similar rules, or if not, what is it that influences the blueprint of the evolution of neural circuits, whether it's like the brain size or it's the neuronal redundancy. So this is basically what they look into, but they start first with a basic background on evolutionary processes. And some of this is quite trivial, and you probably know it since high school, but in my case, it didn't hurt to fresh that knowledge up a bit. And although my high school time isn't that far in the past, I'm not trying to say that anybody is old, but let's just quickly take a look. So we are looking here first at the evolutionary forces and then the evolutionary outcomes. So there are 4 key, or the most central evolutionary forces, which are natural selection. It's basically the addition or removal of alleles harboring mutations. Probably this is something you all know. Then we have mutation and migration and drift. Drift is basically it describes changes in allele frequencies caused by the random sampling that occurs in the production of each generation. And these 4 evolutionary forces are not mutually exclusive, and they can interact with each other as well. So it makes it a little bit more complicated, but also more fun. And looking into the evolutionary outcomes, so there are 3 main types of the phenotypic on the phenotypic level. It's the divergent, convergent, and parallel processes. So the divergent process describes two species evolving from the same ancestor. So we have, the diagrams show it pretty well. So two species evolving from the same ancestor become different from each other. And I think that's like the most prominent thing that people think about when they think about evolution. An example of this is the co-option. It's basically it describes a process where a trait is selected because it gains a function that is different from a function for which it was originally made, or for which it originally evolved. So the benefit of it is that a rapid change, a rapid evolutionary change can occur since the structures are already there, they exist already, and then a little thing is changed. So the example here they provide is in moths. So there is a pre-existing neuron that's co-opted to, like, I think that's the olfactory, it's the olfactory system of the moth, and it co-opted to compensate for their own like wing beating. But that's not, the examples are the next examples are a little bit more interesting. Then we have the convergent evolutionary outcome. It is when starting from ancestral displays do not, when two different when starting from two different ancestors display display in the same behavior. So two different, I'm sorry for my little dugout. So here in the example, we have two frogs which are from two different parts of the world, so South American and Madagascan frog. And they evolved in, or they developed the same maternal care behavior, which is here driven by the same brain part. So it's driven by the preoptic area of the hypothalamus. So the behavior is the same, but the neuronal populations that are active here, which you can see by the different triangles, they are different. So we have two different species basically, which show the same trait. And then looking at the parallel outcome, it's basically that organisms whose ancestors shared a particular trait independently evolve the same trait. As you can see, the example is here with I think flies and mice. So we have basically the same starting points, and the end point of as behavior is the same, but they're still different. To sum it up here, the main ways in which neural circuits evolve are connectivity, neuromodulation. So by connectivity, we mean changes that might occur among just homologous neurons. The neuromodulation, which is basically a change in the expression of neuromodulators, their receptors or degrading enzymes, and neurons, which describes the change to either neuron physiological properties or the change to number and the type of neurons. As I mentioned, I'm going to focus on the connectivity and gonna walk you through some changes in the synaptic connectivity between neurons that have affected evolution, like central neural circuits, and in the end also evolution. Let me scroll to the visualizations. Sorry, it's a long read, but I promise it's interesting. Okay, let's take first a look only at this part. So at the upper part of the visualization. So by connectivity, what they mean is that changes, they mean changes in synaptic connectivity between neurons. So either in the number of synapses or in the type and the number of neurons that are connected. And the author state that it might be just connectivity changes might be the easiest way to modify neural circuits as it allows the conservation of a general blueprint for circuit development. And the modifications that are basically required are very subtle and happen at the last step. So maybe it rings a bell of synaptogenesis. So let's take a closer look at it. They basically reviewed here a couple of examples in different animals, and it's super cool because they you're gonna see it in a moment. So let's start with the divergent network and behavior, which describes a neural circuit that rewired but is producing different behaviors. That first picture A is describing, so they looked into the neural connectivity of certain worm types. So there is like the C. elegans and P. pacificus. So different worm types, different worm species. And it's basically the reconstruction of the neural circuits that is involved in feeding and chemosensation, they say. So it's basically focused on feeding. So because the one of them is a bacterial feeder and the other one is a predatory feeder. And it revealed that while the neuron number and the cell body positions and the axonal branching are conserved, so you can see it basically by the same, by the same neuron number. I think, it's color coded here. While these things are conserved, both circuits have undergone major synaptic rewiring during the evolution. And this is basically an example of divergent evolution of neural circuits through connectivity changes, which leads to divergent behavior because one of them is a bacterial feeder and the other one is a predatory feeder. And the cursor, what you can see, like this little cursor, for example, it shows a certain neuron, which, like in the one species, it's interneurons. In elegans is an interneuron, and in the pacificus, it's a synapse. And this kind of sheds also light on the evolution of connectivity through changes in synaptic specificity. Another example of divergent evolution is this here. It's the sexual circuitry of flies, of the melanogaster flies. This kind of provides some generality to the mechanisms of behavior evolution through rewiring the same or homologous neurons. So the entire diagram shows the sexual circuitry of male flies, let's just call them flies, male flies. And what their behavior looks like is that males evaluate basically the potential partners by tasting some stuff that the females exert. And then they sing by moving their wings to increase female receptivity. How romantic. However, since this behavior is different and highly variable across different fly species, it's ideal to study sensory and also motor circuit component evolution. So there are basically 3 taste pathways that mediate partner acceptance and rejection. It's that's the first one, that's the second one, and that's the third one. So interestingly, what they found out here is that one of the pathways is conceived. That's the PPK23 taste neuron. The other one is presumed to have diverged. That's that one. And the third one, it's here, is the third one conceived, it's conceived in the periphery, but it has still undergone central circuitry changes. So basically oh, I'm sorry that I see that the time is up. No. Okay.
You
super
still
sorry.
have a few minutes, just to let you know, 10 minutes left until 10.
I see. Then let me, I'm sorry that I dived so much into it. Yeah, let me just quickly then explain also the divergent network and the and conserved output. So we know that two species can convergently evolve the same behavior from ancestor that did not display such behavior. And this is basically, they show a neural circuit drift and the convergent evolution in swimming behavior, where they shed light at at basically same having same neurons, but different connections, altered connections. And here again, same neurons, different connections. A super interesting example, I'm so sorry that I didn't have time for it, is the adaptation to changing body size. It shows changes that have evolved via natural selection to kind of maintain the conserved function, although the feature, in this case, the eye size between mouse and rabbit, is changed. So it's basically, it shows how this synapses, how adaptation occurred because of just different body sizes. All right, I'm sorry that I wasn't able to walk you through the entire stuff, but the review is a really good read, and I just recommend to take a look into it, because it's super interesting, but probably beyond the scope of Neuroccino here today.
Thank you so much, and Anna, sorry for slowing your roll here. I just wanted to make sure we have enough time for questions from the people in the room or over on YouTube. First hand is up. I don't know if that's Michelle or Valentina.
Yeah, it's Michelle.
It's actually Valentina.
Oh, it's me. Yeah, I wanted to give you the opportunity to answer the first questions that you described earlier. So do they answer the questions?
So the main answer to all the question is we need more data. Always. But actually, they do. So they look into the sensory periphery, right? Whether that is more evolvable than the central circuits, and indeed they show here with some examples that those things are very likely to evolve together, and there is indeed a bias for the sensory periphery because the central circuits are just less accessible, or have been less accessible throughout the years. But that, like with the new technology, it has been made more accessible, and basically the answer to everything is we need more. We need to study more species. We need to integrate more data. What is super cool, what they actually found regarding the hotspots is, or at least I think it's like one of the coolest things in the whole review, is that they indeed found or were able to show that there seem to be hotspots that are targeted by the evolution. So such as the example with the frogs that come from two continents, and their same maternal care behavior suggests that, or which is also like, which is guided, or which involve the preoptic area of the hippocampus in both species. That's kind of, can be seen as a hotspot. Or here, for example, this P1 neuron, I wasn't able to go that deep into it, but this one is, it basically changes in the connectivity affect the information processing quite a bit. So they were able to answer some of the questions, but not all of them.
And I wanted to add, like, if you look at the panel A, it's fascinating how it's exactly the same neuronal organization, every dot connection between the two species. But the only difference in the functioning of the central nervous system between these two different species is the way it is connected.
And that makes a difference, basically whether it's a bacterial feeder or a predatory feeder, which is super cool. And also the if you look at the muscle connections, you can see that the muscles are in the predatory feeder, the muscles are more connected, which also gives a kind of insight into, or it makes the link, right? So muscles are the motor circuits are more connected, and which perhaps allows the worm Pacificus to be a predatory feeder.
Well, they
Does it make sense?
also that like the function is coming from the way the neurons are connected.
Yeah, exactly.
Yeah.
So yeah.
That's cool. Very cool.
Right. There is
Really cool.
another question from Victor.
Hey, yeah, it looks like a really, really big, interesting article. It's nice that you could like introduce it to us. And I was just wondering, do they talk about like artificial neural networks that are trying to replicate? Not at all. No, it's purely biology.
No, it's really very much into biology. And when it comes then to here, the neuromodulation, then it's even more biological. That are the examples of neuromodulation. And when it comes to neurons, it becomes even more like genetic kind of. They really focus on genetics and on biology. So there is, they do not do anything with, they don't mention artificial.
Yeah, okay, because I was thinking about how like synaptic, or not necessarily synaptic, but network organization in the brains or a nervous system of different animals through convergent evolution can look somewhat like each other. And that it's kind of what some teams are trying to replicate with artificial neural networks in the way that they say, okay, we'll try to replicate, for example, the visual stream in the brain using neural networks that are somewhat similar to what we have, and then we try to let them learn how to recognize stuff, and then we look at them and see if it converged actually, kind of like an actual neural network. And then this, I thought it would have been a nice parallel between natural convergent evolution and artificial silica-based artificial convergent evolution. I don't know if we can call it evolution, but I think the parallel would have been interesting. But if they don't talk about it, yeah, too bad.
No, here they really focus on everything that is basically not artificial and the state of the art in just natural neural circuitry. But super interesting thought.
So you mentioned that they say one of the ways forward is to have more data, more species. Do the authors suggest a way to achieve that? Or is it very much a, hey, community, go out there and collect more data?
Well, so they don't propose a way to, how to do it. But in the very beginning, which is quite cool as well, that's why it's so good read, because it gives you a lot of background information to understand everything. So here, on the first page, basically, they give you an overview of to see what choices drive the selection of which species to study. So that's quite interesting to look into it. They do not say, they didn't have a master plan for getting more data, but the argument is basically look at how much we have already found by studying mice, by studying other organisms, and their argument is to carry on exactly that and to have cross-species comparisons and cross-species studies.
Great. Okay. Thank you. We are running out of time. So I just quickly wanted to remind you that this week, Thursday, we have our final CNS talk for 2022, and it's going to be on finding the network balance in Parkinson's hallucinations. So please do join us on Thursday, 4 p.m. Paris time for the final CNS talk. With that, thank you very much, Anna, for the run through a very dense paper. Thanks for preparing it. We have one more Neuroccino next week, and then we're going to go into a Christmas break already. So do join us this Thursday and next Monday. Have an exciting week in science. Bye.
Thank you.